Good morning, good afternoon, good evening to you all. Before I start with the presentation, I'd like to thank ISA Secretariat, COMRA, and the Hong Kong University of Science and Technology for convening this workshop and for inviting the kiosk for participation. I am Yeon from Korea Institute of Ocean Science and Technology, and I'll be presenting some of the results of the environmental baseline studies conducted by the kiosk scientists in the Indian Ocean over the past 5 years and introduce future plans for the next couple of years. The exploration contract for polymetallic sulfides in Indian Ocean was granted in June 2014 by the ISA for the Republic of Korea, which is the sponsoring state of the Ministry of Oceans and Fisheries, and the exploration began by kiosk thereafter. This is the map of the licensed area of Korea in Indian Ocean, showing 8 clusters with a total area of 10 kilometers squared. On the right-hand side is the research vessel Isabu that was used in most Indian Ocean cruises that we carried out so far. And below is the RV Onnuri, which is another option depending on the availability of the Isabu. And here are the exploration equipment that are used or will be used by the kiosk during the Indian Ocean cruises, such as ROV, deep sea camera platform for the visual survey, and CTD and plume recorder for water column and plume survey. And below are the geological and biological sampling equipment, such as TV monitored grab, box corer, multiple corer, and sediment trap. And I will jump right into the results. This is an example of the water column survey conducted in 2018. And on the left, you see the map showing a total of 12 sites where the water samples are collected using the CTD carousel system, and they are indicated by the black dots. The baseline parameters such as temperature, salinity, dissolved oxygen, pH, PCO2, different nutrients, TOC, metals, REEs, ATP, PON, POC, and methane concentrations are measured from different depths that range from 0 to 4,000 meters. The goal was to see how the water column properties vary with latitude and depth, and how vents might have a control over these variation. The one that showed most significant variation was the methane concentration that we see in the lower right, and this is affected by the vent plumes. And we also look at the vertical distribution of the zooplankton biomass, abundance, and their composition at different sites. In 2018, we sampled plankton using mock nets, net system, or the multiple opening closing net environmental sensing system from the two sites, MOIR 1801 and MOIR 1802, indicated by the blue axis on the map. And the mock nest is the one you see in the figure on the bottom left. This gear allows you to sample plankton at specific size, and you can have a control over where and when to take the samples. And these two sites are approximately 380 kilometers apart from each other. And we did see some variation in zooplankton biomass between sites and depths. As you can see in the first figure, for example, we see high zooplankton biomass, particularly in the upper 200 meters depth, which is typical in euphotic zone in the open ocean. The second figure shows the zooplankton abundance. The abundance was higher at MOIR 1801 site than the MOIR 1802. And this may have been related to the chlorophyll A concentration, which is known as their potential food source. And we actually measured the chlorophyll A concentration, and it was higher in the 1801 than the 1802. And this may also have been related to the current system in the Indian Ocean, and this area may have been affected by the upwelling, which increases the nutrient availability And in terms of composition in the third figure, we saw a total of 97 taxa of zooplankton in the area, and the copepods were the most dominant at all depths, accounting for more than 60% of the relative abundance of zooplankton. The copepods abundance generally increased with water depth, and there were some species that appeared at some specific depths, such as Metridia species or Neocalanus species that appeared only below 500 meters layer. In the next slide, we compared zooplankton biomass and abundance spatially. The sampling was carried out near cluster 1 through 5 in 2018 and 2019. On the right, we plotted the biomass in red and abundance in blue for those that are collected with conical net from the upper 200 meters. We did see some variations spatially. For instance, the biomass was highest in cluster 3, and whereas the abundance was higher at sites that are slightly north to the cluster 3 or cluster 2. And this spatial variation may be related to the environmental conditions at the timing of sampling, such as the current system, the upwelling in this region, which can affect the nutrient availability. And the variation can also be affected by the timing of sampling, either during the day or night or seasonality. And for the biomass, species composition can also affect the biomass. But with the current data that are sampled only once, it is premature to make an interpretation as the actual biomass and abundance may be underestimated or overestimated. So we will continue to sample from the same sites in the future for comparison, and then we'll be able to say what actually controls these variations. And we also investigated spatial distribution of interstitial meobenthic community in terms of their abundance and composition in the study areas. The sediment core sampled from 5 stations were obtained, and they are indicated by the red cross on the map. And we found 5 different taxa of meobenthos, such as nematodes, hypracticoids, and polychaetes, and nauplius, and ostracods. Nematodes were the most abundant group, followed by hypracticoids, copenoids, and polychaetes. And there were some spatial variation in their abundance between sites. And when we look at the vertical profile of the meobenthic abundance in the sediment core, more than 95% of the meobenthic organisms were occurring in the top 2 centimeters at all stations. We also made a list of megafaunal community at the study site. We used deep sea tow camera system to survey this. A total of 98 kinds of megafauna were identified from 16,382 photos that were taken from 16 deep sea camera surveys. This figure is the relative composition of megafauna from 16 surveys. You can see that the composition changes across sites, and on average, the Echinodermata was the most representative phylum, accounting for 51%, followed by Cnidarians, which accounted for 17%, and Chordata and Anthropoda for the next. And in the future, we will examine the relationship between the habitat properties with the community structure changes. And 4 new hydrothermal vent fields were discovered on the Central Indian Ridge. Among them is the Onnuri vent field on segment 3. This is the video taken from the OVF. It is a low-temperature diffusive vent hosted by ultramafic rock with high dissolved methane and low dissolved metal concentrations, and only very small or absent particle anomalies. OVF is characterized by abundant vent organisms forming several clusters within the vent field. In Kim et al. 2020 paper, 21 macrofauna taxa were reported, including hairy snails, mussels, barnacles, shrimps, and crabs. Among the identified 21 macrofaunal taxa, 6 species were not documented at previously known vent fills in the Indian Ocean. And another cruise has been conducted in 2019, and additional species will be reported soon. There are new species discovered from the Indian Ocean, and some have been reported recently, such as bivalve mussel gigantida species from the OVF. This is the first time to report gigantida species from the Indian Ocean. And another new species is the sessile vent barnacle from solitaire hydrothermal vent. This new species is not only the first record of the sessile vent barnacle from outside of the Pacific Ocean, but is also the first sessile barnacle from the Indian Ocean. We have found some new shrimp species from the OVF that are Rimicaris and Albinocaris species that will be reported soon. And we also look at the food web structure, which tells us about the source and flow of energy in biological community, and it is linked to the ecosystem function and resilience that are essential in environmental assessment and management. The food web structure can be assessed based on stable isotope compositions, and we can identify different carbon fixing pathways using carbon stable isotopes on the x-axis and N cycling or nitrogen cycling and trophic position using the nitrogen stable isotopes on the y-axis. What's interesting is that these are the vent fauna collected from one site, but you can see that there is a wide range of values indicating that they utilize a variety source of organic matter. For example, there are bacteria that utilize sulfur to fix carbon, and others utilize methane to produce energy. And based on these results, we can infer the feeding strategies used by the vent fauna for mutual survival and the strength of the feeding relationship that are strongly related to the ecosystem stability. And this is a cluster analysis and similarity profile permutation tests uh, that carried out to categorize the macrofaunal similarity matrix between bent fills on the Indian Ocean ridges, namely Dodo, Longji, OVF, Edmond, Solitaire, and Kyrie. And this is based on the presence absence data and group average linkage, and connected red lines indicate the same group. The results indicate similarity less than 40% between the OVF and other vent fills in the CIR. As of now, only 4 species were reported at Dodo, and as we update the bent faunal composition in the Indian Ocean bent fields, accuracy of the spatial composition or spatial comparison and understanding of the biodiversity will improve. And this is our future plan for the next year to 2024. And we will install moorings with current meters and sediment traps. And we will continue with our visual survey using deep sea camera and ROV and actual sampling of vent fauna to understand the biodiversity and biogeography. And we will also look at the bacterial diversity and trace element concentration in different geological and biological samples, and continue with the water column survey to understand the physical, chemical, and biological properties. And so although Kyoto scientists are trying our best to fulfill and accomplish different areas of environmental baseline studies as much as possible, there are definitely certain areas that are challenging to do without the help of other institutes. An example of those areas would be gene connectivity and biogeography. So we are open and we are willing to have international research collaboration with other international institutes and contractors and we are willing to share genetic information or even sharing of samples. But of course, we would have to ask permission from the government or the individual researchers. But this is something that we can discuss further. And maybe our suggestion is to create some connection or contact points among the contractors or scientists in the Indian Ocean who are studying in the Indian Ocean, that aims for further research collaboration for different topics, including biodiversity and ecology in the Indian Ocean. And this is all I have. And thank you for listening and attention.